Next one is hairy root disease of agrobacterium rhizogenes, that is rhiplasmid. Agrobacterium rhizogenes can also infect plants, but this results in hairy roots and not crown galls as in the case with the agrobacterium tumefaciens. The plasmids of agrobacterium rhizogenes have been isolated and characterized. These plasmids referred to as rhiplasmid. Rhi means root inducing. Rhiplasmids are of different types. Some of the rhiplasmid strains possesses genes that are homologous to plasmids. Example, auxin biosynthetic genes. Instead of virulence genes, rhiplasmids contains a series of open reading frames on the tDNA. The products of these genes are involved in the metabolism of plant growth regulators, which gets sensitized to auxin and leads to root formation. Vector of agrobacterium rhizogenes. As it is done with agrobacterium tumefaciens, vectors can be constructed by using agrobacterium rhizogenes. These vectors are alternate strategies for gene transfer. However, employment of agrobacterium rhizogene based vectors for plant transformation is not common since more efficient systems of agrobacterium tumefaciens have been developed. Importance of hairy roots. Hairy roots can be cultured in vitro and thus are important in plant biotechnology. Hairy root systems are useful for the production of secondary metabolites, particularly pharmaceutical proteins. Plasmid derived vector system. Next one is plasmid derived vector system. The ability of plasmid of agrobacterium to genetically transform plants have been described. It is possible to insert a desired DNA sequence into the tDNA region and then use agrobacterium tumefaciens to deliver this gene into the genome of the plant cell. In this process, plasmid serves as natural vectors. However, there are several limitations to use plasmids directly as cloning vectors. First one, plasmids are large in size. Smaller vectors are preferred for recombinant experiments. For this reason, large segments of DNA of plasmid not essential for cloning must be removed. And second, absence of unique restriction enzyme sites on plasmid. And the phytohormones, that is auxin and cytokinin produced, prevent the plant cells being regenerated into plants. Therefore, auxin and cytokinin genes must be removed. And open production in transformed plant cells lowers the plant yield. Therefore, open synthesizing genes which are of no use to plant should be removed. And open production in transformed plant cell lowers the plant yields. Therefore, open synthesizing genes which are of no use to plant should be removed. Tie plasmids cannot replicate in Escherichia coli. This limits their utility as Escherichia coli is widely used in recombinant experiments. An alternate arrangement is to add an origin of replication to tie plasmid that allows the plasmid to replicate in Escherichia coli. Consider the above limitation. Tie plasmid based vectors with suitable modification have been constructed. These vectors are mainly composed of the following components: the right border sequence of tDNA, which is absolutely required for tDNA integration into plant cell DNA, and a multiple cloning site that promotes the insertion of cloned gene into the region between tDNA borders. An origin of DNA replication that allows the plasmids to multiply in Escherichia coli. A selectable marker gene for appropriate selection of the transformed cells. Thank you.